This is a T cell seen with the aid of time-lapse microphotography. When an organ transplantation is carried out, immediately following the operation, the donated organ must confront the effects of the recipient's immune response. The T cells play a major role in this process. In this film, we will be able to observe various activities of the T cells from both macro and micro perspectives, and to see the effects of the immunosuppressant drug tacrolimus. First, we investigated the effectiveness of tacrolimus in a heart transplantation experiment. A heterotopic heart transplantation was carried out intraperitoneally using a strain of rat with a completely different major histocompatibility complex. The ascending aorta of the graft was anastomosed to the recipient's abdominal aorta, and the pulmonary artery of the graft was anastomosed to the recipient's inferior vena cava. Here is the heart on the third day after transplantation, without the use of tacrolimus. At this stage, we can just see the heart beat faintly. This shows the heart on the 7th day after transplantation. Here we can see that the abdominal area as a whole is moving along with the breathing of the recipient. However, on account of the rejection reaction, the heartbeat has almost stopped, and we can say that heart functioning has almost ceased. This is a view of the heart on the 7th day after transplantation, where a dose of 0.1 milligrams per kilogram of the immunosuppressant agent tacrolimus was administered daily. We can see the heartbeat is maintained. Here is a comparison of the rejection reaction both with and without the administration of tacrolimus on the 3rd and 7th days. On the 3rd day there is almost no difference, but on the 7th day we can see that tacrolimus has had a definite effect. These are tissue samples showing the grafts on the 3rd day after transplantation. In the case where immune suppression was not carried out, we can see that already lymphocytes are invading and a typical acute rejection reaction is taking place. On the other hand, in the example on the right, where tacrolimus was used, almost no lymphocyte invasion is observed, and no myocardial cell destruction is found either. Investigation of the effect of tacrolimus was also made in the transplantation of the small intestine of a rat. The donor aorta and portal vein were anastomosed to the recipient's common carotid artery and external jugular vein. As one example of a rejection reaction in a small intestine transplant, let us take a look at the villi. These are normal villi. We can clearly see the blood circulation in the villi capillaries. Here we see the villi on the 7th day after transplantation. A rejection reaction has occurred, and we can see that normal functioning and circulation have been lost. Next, we see the results after the same 7 days interval, but with the administration of tacrolimus. In contrast with the non-administered case, here the condition of the villi is maintained, and blood circulation is sustained as well. In this image using scanning electron micrography, we can compare the condition of the villi on the 5th day after transplantation. In the case in which the rejection reaction has proceeded, we can see that the villi have become sparse due to ischemia, and the normal condition has been lost. In contrast, in the group which used tacrolimus, the condition of the villi is well preserved. The effectiveness of tacrolimus in transplantation of the small intestine is also confirmed. Today, the progress in the development of immunosuppressant agents, such as tacrolimus, has played an important part in the increasing use of organ transplantation in clinical medicine. In the immune reaction, a T cell recognizes an antigen and an attack is brought about. In carrying out this function, there are two types of T cells which we will discuss: the helper T cells and the killer T cells. Furthermore, helper T cells are classified into two main types: type 1 helper T cells, which assist in the cellular immune response, and type 2 helper T cells, which assist in the humoral immune response. This is a clone of a type 1 helper T cell. When activated by antigen stimulation, various cytokines are released, and the proliferation of other lymphocytes can accelerate. A representative of the cytokines, interleukin 2, is released. Now let us take a look at the reaction in which activated type 1 T cells activate resting T cells. In the same Petri dish, the small round-shaped resting T cells are mixed with activated T cells and cultured. In doing so, as the resting cells come into contact with the activated T cells, they grow larger, and their motility also increases. In this process, activated killer T cells are involved. This is an activated killer T cell. In the cell material, we can see the presence of numerous granules, which were not found in the resting cells. Among these granules are the cytolytic molecules, which destroy target cells. The killer T cells release these cytolytic molecules, which destroy the target cells. In the case of transplantation, this will destroy the graft. In an organ transplantation, the first cells to be targeted by the attacking killer T cells are the endothelial cells of the graft. Here, the endothelial cells are growing. But let's take a look at what happens when we add activated killer T cells. The killer T cells attach to the endothelial cells and begin to destroy them. Killer T cells, which have passed through the blood vessels, react with the cells of the grafted organ. In the case of liver transplantation, the hepatocytes are targeted. On the screen now, we can see growing hepatocytes. When these are put in contact with activated killer T cells, the killer T cells immediately begin destroying the hepatocytes. The closely adhering hepatocytes are cut into pieces by the killer T cells. In the end, the hepatocytes are destroyed one after another. What we have seen here is an in vitro model of rejection, but in a living organism also, we know that lymphocytes would invade the transplanted graft, and destruction of the hepatocytes would occur. Tacrolimus is a newly available immunosuppressant drug which can control this immune reaction response. Tacrolimus controls the activation of T cells. On the left of the screen are lymphocytes, which have been activated by helper T cells, as we saw before. Among the small round lymphocytes, the large active cells are steadily increasing. With the addition of tacrolimus, as the right side of the screen shows, the lymphocytes retain their small shape. 24 hours later, this condition continues. Here we can see that the IL-2, which is released by the helper T cells, has been controlled. We can also compare what happens among these cells to the killer T cells. As we observed before, the activated killer T cells contain many granules, but the killer T cells treated with tacrolimus are small, and the granules are few and not evident. Now let us take a look at what happens when we put the killer T cells, disposed of by tacrolimus, in contact with hepatocytes. Even in the same condition as before, where we saw destruction occurring, now with tacrolimus, the killer T cells do not attack the hepatocytes. Here we can see that the killer T cells of the recipient, unless they are activated, cause no tissue injury. In recent years, remarkable progress has been made at the molecular level in explaining the mechanism of IL-2 production from helper T cells. Here we can see the mechanism of antigen recognition by T cells. When antigen recognition takes place, the stimulation becomes an intracellular signal which is transmitted to the DNA, and IL-2 is synthesized. T cell receptors are composed of alpha and beta chains and of CD3 complexes, which are made up of gamma, delta, epsilon, and zeta chains. Furthermore, these chains have a special intracellular structure, which is called ARAM. Under tyrosine phosphorylation in the ARAM, the transmission of the signal is carried out. An antigen-presenting cell presents an antigen signal together with a class II MHC protein to a T cell receptor. This is recognized by alpha and beta chains, and along with this, a T cell receptor cluster is produced. At this time, a helper T cell surface marker protein, CD4, combines with a class II MHC protein on an antigen-presenting cell. When this happens, an LCK kinase linked to a CD4 is activated. At the end of the LCK kinase, a phosphate P bound to tyrosine is dephosphorylated by the protein phosphatase CD45. With the dephosphorylation at this terminal tyrosine residue, LCK kinase obtains autophosphorylating activity, and it phosphorylates another of the tyrosines in the center of the molecule. In this way, the LCK kinase gains the ability to phosphorylate tyrosine in the Aram area of the gamma, delta, epsilon, and zeta chains, and phosphorylates them. Next, the intracellular protein, known as ZAP70, binds to the phosphorylated zeta chain. ZAP70 itself is phosphorylated by LCK kinase and thus activated. In a similar manner, this process also occurs with an enzyme called FIN, as is shown on the left side of the screen. Along with the activation of ZAP70, the intracellular enzyme PLC-gamma-1 is activated, and a signal transduction proceeds from the cellular material to the nucleus. The activated PLC-gamma-1 produces inositol 3-phosphate, IP3, and diacylglycerol, DAG. IP3 causes calcium to be released from the calcium pool within the cell. This calcium activates calcineurin. Calcineurin is an important enzyme in the dephosphorylation of phosphorylated N-FATCP, and by this process, dephosphorylated N-FATCP enters into the nucleus. With the activation of PLC-gamma-1, the DAG, which is produced then, in turn activates the series of the enzyme groups called PKC, RAS, RAF, and MAP kinase. Through this process, in the end, Jun and Phos proteins accumulate in the nucleus. The NFATCP, which has entered the nucleus, binds with Jun and Phos, and then binds to the NFAT site at the transcription regulating region. Here again, a signal from co-stimulatory molecules, such as CD28, also plays an important part. When this happens, activated signals are transmitted along various routes, and thus an active copy of IL-2 is made. However, when tacrolimus is present, it combines with a protein called FKBP12. This complex has the ability to combine with activated calcineurin and to inhibit the dephosphorylating enzyme activation. To accomplish this, an activated signal transduction route is stopped, and the transcription of IL-2 gene is also inhibited. In this manner, finally, the immune reaction is controlled. Progress has been made in the analysis of tacrolimus and its binding with the intracellular receptor FKBP12. There are cis forms and trans forms of tacrolimus, and within the molecular structure there are differences in the arrangement of C2, N7, C8, C9. In liquid state, the cis forms predominate, but in combination with FKBP12, they become trans forms. This is the three-dimensional structure of the trans form. Here we can see FKBP12. The amino terminal of FKBP12 is indicated by N, and the carboxyl terminal by C. Tacrolimus approaches FKBP12 as if entering a pocket. Within this complex, the part indicated in red participates in the binding with calcineurin. This binding of calcineurin, whose enzyme activity is thus inhibited, suppresses the activation of T cells. With the discovery of tacrolimus, the process of intracellular signal transduction has been further elucidated. It opens a new door in the understanding of molecular biology. Not only does tacrolimus help support the success of transplantation operations in the clinical field, but also the basic research is contributing to progress in the study of transplantation immunology.